PMN310 is a humanized monoclonal antibody against the misfolded toxic form of amyloid-beta, those little clumps, the toxic oligomers. So it's part of this first wave of using passive immunization, so giving the antibodies to the patients to provide protection against A-beta. There are several A-beta antibodies in the works. One is approved from Biogen, Aducanumab, and then there are others in clinical trials, or for us, PMN310, we predict entering the clinic at the end of this year or early next year in a phase 1 trial. The difference from all of the others is taking advantage of the platform that I was describing, where we were able to raise this monoclonal antibody against an epitope that is only present on misfolded toxic A-beta oligomers. So we know that this antibody does not bind monomers, the normal form of A-beta. We don't want to interfere with that. And also does not bind plaque. Plaque is very prominent in Alzheimer's disease, but the thinking has been evolving that it's not the major toxic species in Alzheimer's disease, that it's actually those small soluble oligomers that are toxic to the neurons and are able to cause misfolding of more A-beta, so you have this domino effect of propagation of disease throughout the brain. So the antibody is very selective for that form. Does not bind monomers, does not bind plaque, only the toxic oligomers. And so this would allow us to really, when you dose the antibody, have the entire dose be focused on the oligomers. And by not binding plaque, we also would avoid some of the side effects that are associated with antibodies that do bind plaque. There's a side effect called ARIA, which is imaging abnormalities, brain swelling, microhemorrhages that has been observed in the trials with those antibodies that bind plaque. So we should be able to avoid that since we don't bind plaque, and be potentially more effective, more safe.